The overall goal of this procedure is to allow an experimenter to generate an array of visual stimuli using the VisualEyes system to conduct an oculomotor study. First, the VisualEyes system needs to be calibrated by converting pixel values to degrees using measured targets. The second step is to define the initial and final positions of the left and right eye stimuli by saving the stimuli into a library prior to the experimental session. And finally, the eye movement monitor is placed on the subject and the experimental script is run to record eye movement responses. Ultimately, results can be obtained that show position and velocity traces of eye movements.
The main advantage of this technique over existing methods such as proprietary software is that the operator can program any visual stimuli to conduct any types of oculomotor experiments.
Demonstrating this procedure is a doctoral student, Eun H from my laboratory.
Any type of eye movement monitor can be used for this system. In this demonstration, a limbus tracking and a video monitoring eye movement system will be used. For tandem tracking movements, such as saccadic or smooth pursuit, a single computer can be used for the visual display. To study opposing eye movements such as vergence or the interaction of vergence with tandem version movements, a haploscope is needed with 2 computer monitors for visual display. First, calibration is needed to convert one set of metrics into another because eye movements are typically indicated in degrees of rotation, while computer monitors use pixel values. One can use trigonometry to calculate where to place the physical targets to calibrate the visual displays. For example, if the stimulus on the computer screen aligns with a 2-degree physical target, then that pixel value corresponds to a 2-degree stimulus. To calibrate the system, the operator needs to open the pixel to degree